Hello, everyone. Welcome to Wednesday of week 6. We're continuing where we left off, talking about mass spec, focusing today on de novo peptide sequencing. I hope you didn't accidentally show up to the lecture hall, but I do hope that you get the time to go through these slides on your own, since it's going to be really useful for when you do the second part of Lab 6 tomorrow. So today we're mostly going to focus on some examples for how you might determine the sequence of a peptide given its MS2 spectrum. So for today, first we're going to backtrack a little bit though and talk about some instructions for the week 7 assignment, which is going to be literature review. We'll briefly review tandem mass spec, which we went over for the first time on Monday, and then we'll show some examples for how you might compute the tandem mass spec spectra for a certain peptide and how that's going to help you go and convert from a given spectra to what was the peptide sequence that was actually used to get that spectra, which is going to be your assignment for tomorrow's lab. So week 7 is going to be a literature review. We talked briefly about how that's going to work on Monday. Here is that information again. So just briefly, Monday, May 14th, again, there's going to be no lecture. You're going to have an assignment that I'll go more in detail on the next slide, where you're going to have to choose a paper from one of the tutorials and go over that paper and get a list of questions. On Tuesday, you're going to form groups to discuss each of these papers as a group in your lab sessions and start assigning different pieces of that paper that you'll each be responsible for presenting at the Thursday session. On Wednesday, we'll resume our lectures. Wednesday's topic is going to be some helpful things for when you're trying to go through these primary literature papers. So how you can go through these papers and find out how to get the raw data or find the tools that they were using and learn how to install those. Those are really, really important skills in bioinformatics. They're not necessarily scientific skills, but kind of these practical skills is what's really going to make a difference to you when you're going through and doing your own research. It's really important to be able to get a hold of the data and the tools that you need to be able to work on your different projects. Thursday, you'll meet back up in your lab sessions, where you'll each present your section to the group and use that to fill in a joint Google Doc for your group. And the second half of Thursday, hopefully you'll get a chance to explore the data and the tools for each paper and learn how those relate to the data that we worked with in the lab assignments. For the lab report, there's going to be a one-page review of the article due at the regular time that lab reports are due, so on the following Tuesday. A GitHub link is going to be sent out as usual for this assignment that's going to have all of the gory details about everything that you'll need to do. But for now, some heads up on what's happening on Monday. Again, no lecture on Monday, but you will be responsible for working on this assignment on your own time. We hope this should take you only about an hour, so it should fit into the regular scheduled lecture time. And all of these details are going to be posted on the GitHub Week 7 assignment, as well as I'm going over them here. So we're going to post the papers that were used for each of the 6 different lab assignments. You'll be responsible for choosing one of those papers that you'd like to focus on for the week. So that's part 1 of the assignment is choosing a paper and notifying us on the Google spreadsheet that we'll send out what paper you chose. That way, by Monday night or Tuesday morning, your TAs can go through that spreadsheet and give you different group assignments that you'll be working with in the lab on Tuesday. So you'll need to read that paper and prepare 10 questions about that paper. Those are going to be done through the GitHub link. Again, all of these details will be sent out on the GitHub assignment shortly, hopefully in the next day or two. So that's Monday's assignment. Choose a paper, let your TAs know about the paper in this Google spreadsheet, and write down 10 questions that you have about that paper that you'd like to discuss with your group. Okay, so here is just a preview of what those papers were. These are papers where the data for each of our 6 different lab assignments was drawn from. So week 1 was about antibiotic resistance. Week 2, we talked about mutations in flu strains that would help the flu evade the flu vaccine. So we talked about these hemagglutinin mutations. Week 3, we did de novo assembly. So this paper is about one example de novo assembler. Week 4 was why don't snakes have legs? For that, there's actually 2 papers you could choose. The first one here on the left was mostly about the RNA sequencing data, and the second paper here is where information about the different snakes and how the ZRS enhancer evolved under different lineages. So you could choose either of those 2 if you choose to work on week 4. Week 5 was about eye color. So the paper for that describes the model for how you can use those 6 SNPs to look at eye color. So how they came up with that model, what data they used, and also looking at eye color distributions in different populations. And finally, week 6 is proteomics. So if you recall from yesterday's lab, you took mass spec data from proteins associated with telomeres and used that mass spec data to identify what exactly those proteins were. Okay, so you'll be able to choose any of those. I guess it's actually 7 papers because there's 2 for week 4. So you'll choose one of those for your Monday assignment where you'll read it and write down those 10 questions. Okay, let's first review tandem mass spec, and then we'll go through a short example of how you might actually compute these spectra for different peptides. So if you recall, for tandem mass spec, we had 2 different rounds of mass spec. In the first, we took a protein, we broke, or a complex mixture of proteins for that. We took that mixture of proteins and we broke it up into little pieces called peptides, usually using an enzyme called trypsin that would cut these proteins either after lysine residue, so that's K, or after arginine, that's R. We break up our proteins into these little trypsinized peptides. We do 1 round of mass spec, where we get out this list of, for each mass to charge ratio, we get a relative abundance of the different peptide species. Then if we're doing tandem mass spec, we can take the most abundant ions, or what we call the precursor ions from the first round, further fragment those individual peptides into little pieces using a process called CID, or collision-induced dissociation, and use those smaller pieces for a second round of mass spec. All right, so then that second round gives us more detailed information about what were those most abundant peptides from the first level of the mass spec analysis, and then we can then get actually amino acid level detail about these individual peptides. All right, so that second level we often call the level 2 spectra. And the first level, if you see level 1 spectra, that usually means after that first round of mass spec. Okay, so I wanted to reiterate this process using a nice animation. This credit goes to Jeremy Watrous, who provided this animation about tandem mass spec, which I think hopefully makes this process very clear. So when we're doing mass spec, we again start off with this complex mixture of peptides, or these proteins that were broken down into these peptides. In the first round of mass spec, we take that mixture and we separate things by their mass to charge ratio. And here we can just assume that the charge was 1 and just think about separating things by mass. So from the least heaviest to the most heavy. All right. If these were all equal abundance, we'd get these little spikes at these different mass to charge values. Often these things are different abundance, and so the spectra will give you both the different mass to charge ratios, but also the relative abundance of each of these. Okay, so that's the first round of mass spec. If we're doing tandem mass spec, in that first round, we'll isolate certain precursor ions that we're going to move to the next round. Usually we would isolate the most abundant ions that we're interested in and use those for the next round of analysis. So we would isolate one of these precursor ions, now do a different type of fragmentation, the CID, which we can sort of think about taking this sort of sledgehammer and breaking this peptide into these individual pieces. Then we could do another round of mass spec on those smaller pieces, again getting the mass to charge ratio and the relative abundance of each particle. And here, in an ideal case, each of these different pairs of peaks corresponds to a shift induced by a single amino acid. All right, so we should be able to take this spectrum from the second round of mass spec, read off these shifts between the peaks to directly determine what was the sequence of amino acids that this peptide consisted of. Okay? All right, so our goal in the tandem mass spec is how can we go from the spectra that we got out of the mass spec to identify the peptide sequences behind those, but also what protein those came from. And so hopefully you got a chance to look at the required reading for this week, and you saw that they mentioned there 3 different approaches you might use for doing this. So in one approach, you can do database searching, and we talked about this a little bit on Monday. And so hopefully you guys can see this pointer here. We took our peptide, we did tandem mass spec, and we got this experimental spectrum here. Well, we can also get a database of all the possible proteins in whatever organism we're working with. In our lab, we're working with mouse data, so let's just say we got a database of all the possible amino acid sequences of proteins in mice. We can apply, in silico, we can apply trypsinization, where we just break up those different peptides at every lysine or arginine. And for each of those theoretical peptides, we can compute what the theoretical spectrum would be. This is going to be the topic of the last part of today's lecture, so you'll see exactly how this is done. For any given peptide, we can compute theoretically what should this spectrum look like. Now we can take each of our real observed spectra and just compare it to things in our protein sequence database of our list of theoretical spectra from things in that database, and rank the list of matches. So for each possible peptide, we can get some sort of quantitative score, which we're not really talking about how we would score these, but you can imagine matching up these 2 sets of spectra to get a similarity score between them and ranking them. And then we can assign our peptide to the best match and output some confidence value with that. So that's the database approach. A different approach is de novo peptide sequencing, where we want to go from the spectrum to the sequence, but we're not going to use any additional outside information like databases. So in this type of approach, you go from the spectrum to the sequence only using our knowledge about the masses of different amino acids. Right? So we're not using any sort of database. We're going just from the raw spectra data to the peptide sequence. This is called de novo peptide sequencing. And this is going to be the topic of tomorrow's lab. You can also have a third type of approach where you do a hybrid approach where you combine both looking at databases of possible peptides, but also de novo approach to use those 2 together to try and further improve your predictions of the peptide sequences. All right, so here is a slide again about this review of the mass spec process. Hopefully we now all have a better idea of what goes on during this process. So now we can focus on how we can go from these level 2 spectra back to what the peptide sequences should be. But in order to do that, it's important to have a good understanding of this part of the process I highlighted in red, which is this CID process by which we fragment these peptides into these smaller pieces. Right? Because if we can understand what each of those smaller pieces corresponds to in terms of their masses, and so where they fall on these spectra, well, then we can use that information to go back and reconstruct the peptide sequence. So if you recall from Monday, we talked about this process of CID used to fragment the different peptides. We usually think about for each peptide, it would get broken just at one place in the middle. So every time you break this peptide, you're going to end up with 2 different smaller pieces. The part of that original peptide that extends from the N terminus, so that's the part on the left here, that we refer to as the B ion. The part extending from the C terminus, so the part over on the right, is referred to as the Y ion. Right, and you can get a different B ion versus Y ion for every possible break like this between the amino acids. And the resulting spectra that we see is actually a mixture of these two different types of peaks, both the Y peaks and the B peaks. All right, so here's just an example peptide for the different breaks you can get. So if we had this example peptide shown in orange and we applied CID, notice again that this peptide ends in lysine. These are almost always going to end in a lysine or an arginine, since they're all what we call triptych fragments, or they resulted from trypsinizing a bigger protein to start with. All right, so we take this fragment and we apply CID, so we can get every possible break along this peptide, which gives us this collection of different possible ions. Those ones extending from the left or the N terminus are the B ions, and from the right, from the C terminus, those are the Y ions. Right, and again, when we apply the CID, we get our Y ions and our B ions, and what we see as a result in the spectra is a combination of the different peaks for each of these different possible ions. And if you were to read off between the different peaks from left to right, you get the B ion series, gives you, and these are in blue, you can theoretically read off the different amino acids by looking at the shift between these two pairs of peaks, seeing that corresponds to B. The shift between the next two peaks corresponds to the mass of C, and so on, up to the final mass here on the right, which is going to be the total mass of this peptide, or the mass of the precursor ion that we put into the second round of mass spec after isolating it from round one. Okay, so the B ions generally go from left to right. The Y ion series goes in the opposite direction. So the furthest shift to the right over here represents the first amino acid from the N terminus, and then you can read from right to left to read off the amino acids all the way to the C terminus. Okay, so this was a theoretical spectrum here. It looks very clean and nice. Theoretically, you might expect all these different peaks to be at very similar abundances if breaking these peptides using CID was just random. But in reality, what you see for your spectra looks a lot messier, so you might see something like what's shown on the right here. So in reality, there's a lot of non-random things that happen. So for instance, not every possible break for the CID is equally likely. Some places are much more likely to be broken or fragmented than others. Also, different ion types, like Y versus B ions, depending on the original peptide, Y or B ions might be more prevalent than the other type, because maybe one is more likely to be ionized and the other might kind of fall out of our experiment. And we'll see at the end there are kind of all sorts of reasons why these peaks might not fall exactly both at the locations that we expect, but why they also have very different abundances from each other, even though you might have expected a priori that they would have very similar abundances for each peak. Okay, so here's a preview of some complications you can get. Well, for one thing, theoretically these spectra should correspond to the weight of different amino acids, but you can get different modifications to amino acids, such as some of these PTMs like phosphorylation or acetylation we talked about on Monday. You can get the peptide broken at multiple bonds, so you would get something that's not exactly either in the Y series or the B series, but it's something else. That might cause some confusion. And again, not all these peak types are equally likely. Some might be very abundant, and other peaks that you might expect in your spectrum might be completely, totally missing. Right, so it can get kind of complicated. Our goal here is to get an understanding of how this process works, but your you're welcome to read about additional levels of detail that you can get into when trying to reconstruct these sequences. So to help us understand this process more, we're going to go through an example of how you would compute the theoretical spectrum for a given peptide. And I'm pointing you to the link where this example was taken from that gives you much more detail on this process of how both you can compute the theoretical spectrum for a given peptide, but also what we're going to try and do, which is go the other direction. So, from the spectra, compute the peptide. Here we're going to talk about going from the peptide to the theoretical spectrum. And the lab, you're going to go from the spectrum and try and figure out what the peptide was used to generate that. Okay, so here for our example, we're going to compute the theoretical spectrum of this example peptide shown here. And just to start off, this is showing, the bottom picture here is showing the actual spectra for this, which is, as we talked about before, quite messier than what we would expect if we just saw the theoretical peaks. All right, so let's start off with this example peptide. Again, it ends in a lysine because these are usually triptych fragments that we're working with. All right, so we can take that peptide and break it up into the different possible B ions. And if you remember, the B ions extend from the N terminus. So all of these different possible B ions are going to start with G, with glycine, the first amino acid here in this peptide, and they're going to extend from G, going all the way from just the single G all the way to the entire peptide, extending all the way to the K. So these are all the possible B ions that you could get from this peptide. All right, we can compute what the masses of each of those different B ions should be, given prior information about what sort of mass shift we would expect from each possible amino acid. This table is shown here. It's also reproduced in your lab assignment for tomorrow. So it's good to keep this handy as you're going through and computing these different mass shifts and trying to figure out what the peaks correspond to. But I did want to point out a couple of complications looking at this list of masses. So one thing you'll notice is that sometimes a given mass shift might not be completely clear about which amino acid it refers to. So if you look, for instance, at the mass of leucine, let's see, we have 113.08. Well, that's almost identical to the mass of isoleucine. Right, so when we're going through these spectrum, we're not going to be able to tell the difference between those two different amino acids. And you'll see that some of the other ones are also quite similar to each other. For instance, glutamine and lysine have very similar mass shifts as well. Another thing to point out is that you could have combinations of smaller amino acids that when combined equal the mass of a larger amino acid. So for instance, twice times the mass of glycine is approximately equal to the mass of one asparagine. So if you're going through your spectrum, you could have a peak that corresponds to either 2 Gs. That would be the same location as a peak corresponding to 1 N. Which can can be difficult to pull apart, especially if some peaks are missing from your data. So that might be a common mistake, is accidentally thinking that 2 Gs correspond to an N residue when it's actually 2 Gs. Okay, but in general, you'll be able to refer to this table of masses to figure out what shifts each of your peaks corresponds to. So if we go back to our B ions here, we can use this table to compute the mass of each possible B ion. And here we're just zooming in on a subset so we can get some additional detail here. So let's look at this example B ion, this GLS sequence, and we can compute where we might expect a peak for this ion. So we can get that by adding together the mass shift of glycine plus the mass shift of leucine plus the mass shift of serine. And then there's an additional complication here is that we have to add 1 Since there's one extra H here at this G terminus residue that's not present on the other amino acid here, so we have to add this additional one. So we do that same additional one if we were adding the mass of these other peptides as well. All right, so for this example, B ion, we have mass shift of glycine versus plus leucine plus serine plus 1. We get 258.1, which matches what was previously calculated for this ion. So we could calculate these masses for every possible B ion for our peptide. All right, we can go through a similar process looking at Y ions. So here are all the possible Y ions you can get from this peptide. The Y ions extend from the C terminus toward the N terminus. So they're all, in this case, going to start with a lysine and extend towards the N terminus. We can do a similar process where we can compute the mass for every possible Y ion. So let's look at this example where we have a lysine and a glycine, and well, now you're going to make me remember what W corresponds to. Let's say, and we have a W. So again, we can add up the masses of these three different amino acids. There's a couple of extra little complications here. These are all going to have an extra +1 because they have this extra H plus for each of the different Y ions. And they're also going to have an extra +18 because of this extra OH hydroxyl group on the lysine over here. So to get the mass of each of these Y ions, we add up the mass shifts of each of these different amino acids, and +1 for this hydrogen and +18 for this OH. So for this example, we get a total of 390.2, which matches what was shown here for this ion. All right, so you can repeat that process for each of the different Y ions in your series as well. All right, and so we can do this process for every possible B ion and every possible Y ion to get the different masses where we would expect each of the peaks to be when we were to do our tandem mass spec on this specific peptide. Again, this is what the actual spectrum looks like when we do this experimentally, but you can go through and identify many of these peaks correspond to what they should have been theoretically. And we could just spot check a couple of these here. So if I'm looking at, say, the Y series, I see 390. I should have had a 390. I see 489. I see this 489. Let's see, the next one was 603. I see 603. And then one more, I see my 716. Right? And so it looks like in this example, the Y ion series is more prevalent. And we can kind of go through this list of the different locations of these peaks and visually verify that these peaks are actually present over here in our spectrum. Right, so hopefully now it's more clear how you can go theoretically from a given peptide going to what the spectrum should look like. So if you were going to try and go the opposite direction, if you wanted to take this spectra and infer what the peptide actually is, well, that's a little bit harder problem because now you're dealing with kind of all the mess that happens when you're looking at real world data. But we put some clues here on this slide, and you can also refer to that tutorial that was linked to. So I mean, we can go through these different steps. First by following the B ion series. Well, a good thing to look for to start with is the last amino acid, since usually you can bet that that's either a lysine or an arginine. So you can look at the two possibilities and calculate where the corresponding B and Y peaks should have been if the last amino acid was a lysine versus arginine. And that's an easy way to get started with the first one. After you've found those peaks corresponding to either lysine or arginine, you can look for the next, say in this case, B ion in the series. And here we're going to go from left to right. So if we want to find out what the second amino acid is actually for B ions, we're going to go If we started with lysine or arginine, we're over here at the end. So if we want to know what the second to last amino acid was, well, then we can calculate all possible shifts based on the masses from each of our different amino acids and see if for any of those shifts, if we actually see a peak at that corresponding shift in our data as evidence that amino acid is indeed the next one. Once you've figured out that for, say, D and E here, now you can repeat the process going from right to left. So now I'm at this peak here. I can again calculate all possible mass shifts to get me to the next peak and find a next peak that corresponds to one of those mass shifts and read off what the corresponding amino acid should have been. You can do a similar approach for the Y-ion series, where again you can start with hopefully figuring out what the last amino acid was, whether it was an arginine or a lysine, and doing the same process where you calculate going from this time left to right for each possible amino acid shift, where do I see a corresponding peak in my data. So hopefully that gives you an idea of how you might go through these spectra and read off the peptide sequence. There are a lot more details that we could have gotten into this if we had time. We don't have time to go through a lot of those extra details, unfortunately. So I hope that you'll at least in the lab get an idea of how this process works and how it could be used to read off these different peptide sequences. And I just wanted to mention, mostly to give you an idea of how this gets complicated, that many different complications can happen. So we mentioned that some fragmentations are more or less frequent than others. Sometimes of ions can occur more or less frequently than others. There's also, finer grain sort of complications. So for instance, fragments containing K, R, Q, and N may appear to lose that ammonia group. So some of those might have a -17, but not always. Things containing S, T, and E may appear to lose water, so a -18. B ion intensity drops when the next amino acid is PG or HK or R. And sometimes the ion series will kind of just die out if you have an aspartic acid somewhere in there. And so, I don't expect you to memorize this list, but when you are going through your example spectra tomorrow, some of the peaks that you might expect to see are going to be completely missing, and some might have slightly shifted masses compared to what you would have expected. And some of these sorts of rules might be in play there. Again, here our goal is to get an idea of how this process works. And if you're interested, you can go into more detail into that process. All right, so Lab 6. Hopefully yesterday you got a chance to dive into some of the mass spec data for identifying proteins associated with telomeres. For part 2 tomorrow, for the most part, we're going to be taking an example spectrum and trying to figure out what the peptide sequence is behind that. In addition to doing that process manually, we'll use some computational methods that try and automate that process so we can compare our results to what those tools get. All right, thank you for tuning in and listening. I look forward to seeing everybody next week, and I hope you have fun doing Lab 6.